Hello, I would like to present my understanding on the article titled The Effects of Paternal High-Fat Diet Exposure on Offspring Metabolism with Epigenetic Changes in the Mouse Adiponectin and Leptin Gene Promoters. I would like to highlight the importance of this literature by citing a prevalent disease, obesity. It is a public health crisis, and it contributes on a global scale to morbidity and mortality. In fact, 40% of adults in the US alone are obese, and there is equal disease incidence between males and females, regardless of age. If a mom is obese during pregnancy, then that may cause fetal overgrowth, and this increases risk of developing other diseases, namely metabolic syndrome and possibly type 2 diabetes. It increases risk of complications during pregnancy, and it's implicated in the transmission of obesogenic and diabetogenic traits for numerous generations. In fact, a study showed that there's an association between the maternal and offspring BMI for 3 generations. The BMI of the father influenced the offspring BMI both independent of and in addition to maternal BMI. I would like to provide background on the biological system examined, and that would be adipose tissue. That biosynthesizes adipocytokines, leptin and adiponectin, both of which are peptides. Adiponectin is anti-diabetic, anti-atherogenic, and anti-inflammatory. Generally speaking, it regulates glucose levels and fatty acid breakdown. Decreased levels are affiliated with obesity, insulin resistance, and type 2 diabetes. Leptin regulates satiety and energy homeostasis. More generally, it inhibits hunger. It is essential for the development of metabolism, the hypothalamus, and food-seeking and reward behaviors. And by extension, nutrients may influence the methylation of DNA and the modification of histone proteins. Prior research states that mice, which were fed a high-fat diet, can cause a metabolic syndrome-like phenomenon to be passed on to the offspring via epigenetic changes in genes encoding adiponectin and leptin. Obesogenic and diabetogenic traits were eliminated following 3 generations of the mother being fed a normal diet. Malnutrition during pregnancy may cause epigenetic changes, and that could be involved in passage of metabolic diseases for numerous generations. Obesity in the father causes changes in the metabolism for 2 consecutive generations. The objective of this particular project was to ascertain does paternal obesity caused by feeding high-fat diet impact the metabolism of the offspring through changes in adiponectin and leptin genes, and it was also to determine whether normal diet exposure for numerous generations eliminated the effects induced by these epigenetic changes. So what the experimenters did was that they obtained male and female 8-week-old Institute of Cancer Research ICR mice, and they fed these males and females GLIM, high-fat diet or control diet, 6 weeks for males and 4 weeks for females, following which they were weighed and mated. 4 groups for the purpose of the first objective were created: control diet male and control diet female, high-fat diet male and control diet female, control diet male and high-fat diet female, and high-fat diet male and high-fat diet female. For the second objective, 4 groups were again looked at. The offspring from males fed a controlled diet for 0 generations, 1 generation, and 2 generations, following paternal high-fat diet exposure, and the offspring descending from mice which were only fed controlled diet. The triglycerides, adiponectin, and leptin levels were measured from blood samples and immunosorbencies assays, particularly ELISA kits. The results are as follows. The figure on the right, it shows the experimental scheme for the F0 parental generation. So A represents the offspring from the controlled diet male and controlled diet female. B represents the offspring from the high-fat diet male and controlled diet female. C represents the offspring from the controlled diet male and high-fat diet female. And D represents the offspring from high-fat diet male and high-fat diet female. The litter sizes from each group, there was no significant difference. The figures on the right, they show fat mass to body weight ratio as a function of weeks after birth for both females, which are on top, and males, which is on the bottom. The key is open circle for A, square for B, triangle for C, and diamond for D, which is the reverse order of the lines from top to bottom on the graph. Please bear in mind that a star represents a p-value of less than 0.01 versus group A pound sign represents a p-value of less than 0.01 versus group B, a dollar sign represents a p-value of less than 0.01 versus group C. So as you can see, group C and D, the top two lines, they experience a fat mass gain which was significantly more than what group B experienced. For group C, this began as of 12 weeks. For group D, this difference was seen from 8 weeks point. And when group C and D were compared, the fat mass gain in group D, it was significantly more than that of group C as of 8 weeks. The figures on the right represent the concentrations of various species, trisoglycerol, adiponectin, and leptin for all groups A, B, C, D. They're all offspring and they're all 24 weeks of age. So if you see group C and D, their concentrations of trisoglycerol, adiponectin, and leptin, they were significantly different from those of group B. And when both C and D are compared, trisoglycerol, leptin, and adiponectin levels, they were significantly different from group C as of week 24. So the findings of this study was that if the father is exposed to a high-fat diet, that had a weaker effect. It had a lesser effect than did exclusively maternal high-fat diet on the offspring. But if the two are combined, then the paternal exposure to high-fat diet, it had an additive effect on the maternal high-fat diet for 2 generations. These effects, they were eliminated if the male offspring were fed a controlled diet for 2 generations. Now, extending beyond the data I presented, the paternal high-fat diet results in a metabolic syndrome-like phenomenon because of modifications in the genes encoding adiponectin and leptin, and these changes persist for multiple generations. And now these effects, they are reduced if the father is exposed to a controlled diet, when the mother is fed a high-fat diet, and the mother's effects, they are completely abolished following 2 generations of controlled diet consumption by the offspring. And also, independently taken, the paternal high-fat diet effects were less than those of maternal, but when both are taken together, they are additive. I would like to for you to think about these questions. So based on what was presented, what physically do you think occurred to DNA encoding the leptin and adiponectin peptides, and where exactly on the genes? And in what ways do you think this research would be beneficial for humans? Thank you.